Our next speaker is Andrey Klymchenko. He just told me that he even updated his presentation last minute before he came here. So, so probably you had some good ideas last night, which you wanted to include it. So his presentation, imaging integrity of lipid nanocarriers in bloodstream and tumor of living mice. So we are looking forward to your last slide from this morning.
Yeah, thank you, Chairman, and thank you, the organizers. Yeah, so, okay, I can see like this. Okay. Yeah, in our lab, actually, we are working on different type of particles, and, oops. Hopla. Okay, yeah, different type of particles, starting from very small ones, which are protein-sized micellar particles, and then dye-doped polymer nanoparticles. These are a little bit larger, 10 to 18 nanometers, and then even larger objects are lipid particles. And the applications are very different, like super resolution for the small ones, and fluorescence imaging, and then cell tracking and detection of target biomolecules for polymer systems. And lipid-based particles are particularly suitable for in vivo imaging. So here, I will focus on this part. And why lipid nanoparticles, and what is that? So those, in our case, those are nanoemulsions, essentially, which are composed of the oil core and this hairy pegylated surfactant. And the important thing that the components are generally recognized as safe GRAS materials, and the caliphore or chemophore, which is a surfactant, it is FDA-approved injectable material, and it's actually a component of paclitaxel chemotherapy. But the thing is, so instead of drugs, you can put drugs inside or contrast agents. So here I will focus on contrast agents, on fluorescent markers, which will reveal some interesting things about these particles. So how they're made, it's a very simple protocol. This is another advantage of this system, that you actually mix oil and oil surfactant. You heat that at 40 degrees, and then after good mixing, you add water, and spontaneous emulsification produce particles. And the size of these particles depends actually on the ratio of oil and surfactant, so it's that simple. And you can go from 20 nanometers up to almost 200 nanometers. But the thing is that even one article when we published, the reviewer said, these are not particles, these are just droplets. And okay, it's a definition, but it's true that these are liquid systems. How well they encapsulate the cargo molecules, and because their core is liquid. And as our earlier studies, we first naively tried Nile-Red, which is a kind of model drug molecule, which is also lipophilic. And it was binding well the core, but once you inject, for example, in the zebrafish, you see a very nice leakage of this dye everywhere in the tissue. And later on, we showed that actually by using highly hydrophobic molecules like cyanine and this bulky hydrophobic anthrine, which makes ionic molecules very apolar, we can first load much more dye inside, like 40 times more. And you can see, this is the image of zebrafish. You could see the blood circulation exclusively. You don't see any leakage. So it's very important, the hydrophobicity of the system, of the cargo, to preserve inside droplets. So now the key question was, how stable are those droplets in vivo? Because they are essentially liquid systems. So, and if we apply them for drug delivery, they are supposed to bring the cargo molecule up to the tumor. And so we wanted to follow all the way up to tumor whether the cargo will be inside. And for that, the unique approach is so-called FRET, first resonance energy transfer, where we can put two fluorescent dyes together. When they're together, we will see the acceptor emission. And when they get decomposed, we will see only donor emission, because donor and acceptor molecules get separated. So but for that, we need dyes which operate between 700 and 800 nanometer, which is an optical window where the tissues are transparent. So you can use a fluorescence microscope where we can face whole animal imaging. So our system was a couple of near-infrared dyes, cyanin 5.5 and 7.5 with long and kill chains, so again, very hydrophobic. And we needed this magic contrarian to make those guys well soluble because they're ionic, and so we need a contrarian which is very hydrophobic. And so like this, we place these guys together in the droplet at high concentration to produce this energy transfer. And so this is there. First, we have to optimize the formulation. So we change the concentration of our FRET pair. And when we get up to 1%, so we observe decrease of the donor emission and increase of the acceptor emission, which is normal because at high concentration, donor acceptor gets closer to each other, and you have nice FRET. So 1% was chosen as the working concentration. Then we have to check whether our system works. When we make a disruption of the particle by organic solvent, we put dioxane. We observe that initially FRET system with the acceptor emission turns into exclusively donor emission because donor acceptor separates, so the system works. And now, first test, we did stability in serum. So when you add 100% serum, you have only little change in the ratio of the acceptor versus donor, or versus the sum of acceptor donors, it doesn't matter. So the changes are small. Of course, in water, there was no change. So it is stable in serum. So the next thing, when we want to quantify the integrity of the system, we have to calibrate first our device, and so our imaging setup. And for that, also, we have to have a model of the integrity. And for that, so we mixed actually fully 100% integrity particle, which is our 1% FRET pair, and separately donor and acceptor, as supposed to be 0%. So we mix them, and then we check the spectra. As expected, the donor increases when integrity decreases with respect to acceptor. We mix these guys and put them in the vials and do imaging. So this is a donor which is increasing when integrity decreases, and acceptor slightly decreases. Then we make a ratio, and this color actually reflects the integrity of the system. So once we have that, we perform the calibration, where this ratio of the channels reflects the integrity. So then we inject it into mice, and what we could see, actually, that we have a strong acceptor emission, which reflects their FRET in our mice, and especially in the blood circulation, you can see in the tail vein, and you can see also in the liver. So then this color actually is map for the integrity, which you can decode from these colors, actually. Each color, and here the color red, means 100% integrity in the blood circulation, and the rest is already less. So with time, you can see that the donor fluorescence increases and acceptor fluorescence decreases. So what happened actually, that the particles slowly disintegrate. And when you place in the region of interest, like liver, you can see there is a disintegration with a half-life of 8.2 hours. And for instance, in the blood circulation, which we focused actually on the tail vein, there was almost no change. So those nanodroplets are very stable in the blood circulation, but in liver they decompose. The same we did with the xenografted tumor. So it's a mammary carcinoma. And what was nice that these particles accumulated rapidly within 1 hour in the tumor. And then important that the FRET signal was very high, which actually reflected that we have almost 100% integrity once the droplets gets into the tumor. So this was really important to see that those liquid systems can enter tumor in nearly intact form. But with time, we observe the disintegration of the system with a half-life of about 4.4 hours. And of course, we checked whether after dissecting the mice, we could see that it selectively gets into tumor more than into liver. So in conclusion, what I try to present you is their system based on lipids. Those lipids nanodroplets can be made of any size. We can load them with organic molecules, especially with fluorescent dyes, and quite effectively. And then these are components of this system are FDA approved, and we describe the method of imaging based on FRET, which will allow you to quantify the integrity of the nanosystem, and in this case, the lipid. So importantly, those lipid systems can accumulate into tumor. Guess this is by EPR, because we did not have any targeting ligand. And importantly, they disintegrate only after entering the target tumor, so which is important for future application in drug delivery. And so the next natural step is to go for drug delivery with these systems. And so I would like to thank my group, which works hard on different fields, but those projects are carried out mainly by Redwan Ushala, who is present here, and his poster number 8 is here. And then Bogdan Andrey, who synthesized the molecules. Then so the Biophotonics groups who assist us with some, help with us with instrumentation, imaging, and the collaborators, especially Jackie Goetz and Nicolas Anton and Van Damme, and from Strasbourg. And of course, I have to thank the financial support, especially ERC and the others. So thank you for your attention.
Thank you very much for your nice presentation this morning. Must be a lot of questions to you.
I hope so.
Uh-oh.
What's the circulation time of your particles in blood?
Yeah, the circulation time we measured from directly from fluorescence, and through fluorescence intensity we could have about 3 hours half-life circulation, but in fact it is longer. I think we are limited by the signal. I think we have to directly take the blood samples and check, because with CT scan experiments we get 8 hours half-life circulation. But yeah.
This fluorescent images could be very misleading, actually. Your particles could be deposited in the skin, for example. You're just imaging the skin deposition instead of true blood-borne particles. So you have to be aware.
But here actually, in fact, we are quite happy with the quality of those images. With the quality of those images, because you clearly see actually localization in the tail vein, and this signal disappears actually within 6 hours. You don't see anything. It can be seen here. You have here a clear signal. The signal actually doesn't change much in the whole body, and then it disappears in the tail vein. So that's not skin. And when you see skin, it's just a homogeneous fluorescence on the surface. That we can distinguish easily on the image. But of course, we have to be careful with interpretation.
Thank you, Andrey, for a very nice presentation. So how readily is this system transferable, for instance, to liposomes or other nanocarriers, where you can use the FRET principle in order to see disintegration as a principle?
Yeah, I think the key difference is the core, because in liposome the core is water. Here the core is oil. So therefore, in principle, the difference that we can solubilize lipophilic material directly inside the core. And for the FRET experiments in vesicles is done mainly on the membrane. Or if you can encapsulate, then you have to precipitate something inside the liposome. But I would say the quantification of integrity with the liquid system, liquid core, oil core, is very straightforward, because you can calculate precisely how much you have inside and how much has been released here, because it's a simple solution of organic dye inside oil. Yeah, so I don't know if I... Yes, you can use the membrane of liposome, but the problem when you use dyes at the membrane surface, they are too much accessible to the proteins of serum, and you will have a leakage very fast. It's extremely fast. We observe this with membrane probes. It's minutes. But when it's hidden in the core, it's hours or more. Yeah.
Anybody else? If I understood that correctly, I think in the second slide you said that your droplets, call it that way, are pegulated?
Yes. So it's the surface is actually covered by cauliflower, which is one side is a lipophilic part, and you have a peg of about 1,000 molecular weight. So we have it's a well-pegulated shell. Okay. Yeah.
I'm just asking the question. Tomorrow there will be a presentation on pegulation, and that a lot of kappa reactions have been seen if, let's say, nanoparticles or droplets, liposomes are pegulated. Have you looked into that issue already?
In there, kappa reactions?
Kappa reactions, that means, let's say, you get a type of, let's say, IgG reactions, pseudo-IgG reactions?
Yeah, in fact, the Cali-4 is known to have allergic effects. But okay, it is still FDA-approved material compared to many other surfactants which are even more toxic. But we did not check that. I think once we get into the application, real application, for at the imaging stage, the doses are actually very low. Okay. And then when we have to go to delivery, we have to increase like 20 times the dose. So then I think all problems may start.
Yeah, I just came to this point because I know that the FDA just is looking into that and have some new regulations. So we all suffer from, I would say, if you get to the more, let's say, practical clinical point. Okay?
Okay.
Thank you very much.
Thank you.